In the previous video we saw an introduction to dynamic modeling. We will now move on to the most classic example of a biochemical network model, which is Michaelis Menten kinetics, and we will also look at another example system in this video. So, let us look at the classic Michaelis Menten formulation. So, Michaelis Menten formulation is to study the rate of an enzyme, the initial rate of an enzyme catalyzed reaction. So, what is the wiring diagram, if you will? So, this would be the wiring diagram, E plus S right from here the assumptions will start, right. So, you may see in most books that this may be written as a forward reaction, but that is not at all necessary. What is the rate of the reaction? What do we mean by the rate of the enzyme catalyzed reaction? You may all be familiar with this, right? V equals V max into S by Km plus S. What is this V? Rate of what? dP by dt. So, what is dP by dt? Sorry, K2 into ES minus K minus 2 into E into P. This is where the first important assumption for Michaelis Menten comes. People normally eliminate this term. Why?
Because product to intermediate formation is very.
When can you eliminate? When can you? So this can be 0 in 3 scenarios. E equal to 0 is a moot point. We will not even worry about it. So either K minus 2 is 0 or P is 0. So P equals 0 means initial rate, or if we can have the product removed, right, K minus 2 would mean irreversible reaction essentially, but that is a harsher assumption to stay with. Right. So dP by dt is K2 ES, so we will just assume that it is K2 into ES. So what is ds by dt? K minus 1 ES minus K1 ES. dES by dt? dES by dt will be little more complex. So, it is going to be K1 into ES minus K minus 1 into K minus 1 into ES minus K2 into ES. Actually minus this term as well, but we have already chosen to ignore that term. And this is where you apply the, and you have also another equation, right, E plus ES equals constant, in fact equals E naught. So, this is one algebraic equation. You combine it with the rest, you can essentially compute that, and you also have to usually apply one assumption, right. There are two popular assumptions. One is the classic Michaelis Menten assumption, the other is the Briggs-Haldane assumption. What is the Briggs-Haldane assumption? dEs by dt is 0, quasi steady state. Michaelis Menten assumption is basically ds by dt is 0. Based on this you get different values of Km essentially, but the equation remains as dP by dt is K cat into E naught into S by Km plus S. So, this is actually, so K cat is going to be involved in these terms, right. So, K cat is the turnover number, right. So, your V max is actually K cat into E naught. So, what are the parameters here?
K cat, E naught,
K cat and Km. E naught is like an initial condition more or less, it is the concentration of enzyme, but K cat and Km are your parameters. Obviously, units of Km
consider as substrate.
concentration, substrate and K cat will have its own units. K cats will be units of inverse time. And Km micromolar, nanomolar, whatever, some concentration units. So, this is classic Michaelis Menten. You can obviously add layers to this, competitive inhibition, uncompetitive inhibition, irreversible inhibition, substrate inhibition, presence of other compounds and so on and so forth. But the basic idea is there is a simple algorithm to derive the rate equations, and this is not something that you will do on a daily basis, you will just look up a table, right. You do not want to work out the Michaelis Menten carefully in every case, unless there is some special thing that is happening in your system. If you have multiple reactions or things like that, then you may have to worry about it. So, what are the assumptions in Michaelis Menten? So, there are a bunch of fundamental assumptions and a bunch of assumptions that are needed for the Michaelis Menten equation itself, like Briggs Haldane and Michaelis Menten assumptions, but the fundamental assumptions are more important. It is an isothermal system, we have we've neglected the effects of temperature, right. So, and there is enough concentration of all these molecules such that you have started worrying about concentrations rather than molecule numbers. It is not a in the stochastic domain, right. When you have very few numbers of molecules, you have to worry about the probability of collision between enzyme and substrate, whether that even happens. Now you basically start writing in terms of concentration, and of course you assume free diffusion, and you also assume that the cellular space where this happens is somewhat homogeneous. It has there are no spatial gradients of the components and so on. So, there are so many implicit assumptions when you just even wrote the law of mass action. So, remember therefore, it is not, as I would have shown you here, it is not the law of mass action, but it is actually a model, right. So, this is a systematic way to derive the equations. You first draw a wiring diagram and assume mass action kinetics and write out all the equations, and sum of all enzyme containing species is E total, that is one algebraic equation, and then assume quasi steady state for n minus 1 enzyme species, right. So, this would mean ES sorry ES, ESI, EI if I is the inhibitor, whatever are these other species, and then dP by dt is your final reaction rate, which you have to substitute do some basic algebra to arrive at. So, this is the larger Michaelis Menten system where you have all kinds of possible combinations. So, standard Michaelis Menten is just this and this, but if you have competitive inhibition, it is this and EI plus S with a reversible reaction. You can have uncompetitive inhibition or non-competitive inhibition and partial inhibition. There are various ways of various types of inhibition that can happen. Each of these will give rise to different kinetic equations. So, these have been derived in the book by Clip. So, it is a very nice reference to look at. So, you will see that these are all the equations you need to worry about. So, the moment you see this kind of a reaction happening in your system of interest, just plug in one of these equations. So, you may also have substrate inhibition where E plus ES and S bind to form ESS, which cannot form a product, and it is most likely reversible. You can have other ligands bind to the protein, you can have cooperativity. So, how does the Michaelis Menten equation plot look like? Right. So, initially it goes like this and it goes to saturation. Half of Vmax. What is the shape of this curve? What is a rectangular hyperbola? What is the canonical equation for a rectangular hyperbola? xy equal to c, or rather you can see that you can write the Michaelis Menten equation in that form. Try it out. More interesting, very commonly in biology, you have the Hill equation, which can look like this when there is no cooperativity, right. So, this comes from the equation V equals V max into S by Km S, except you have a different Km and you have an exponent. N equals to 1, classic Michaelis Menten kinetics, n greater than 1, cooperativity. So, how does it look? It is difficult to draw these nicely. Something like this, right. So, as k increases, as n increases,
It will be like a binary.
it becomes more switch like.
Step.
It becomes like a step, right. It becomes more switch like. Right, for high n switch like behavior, right. But what is it that you actually see on the on this curve, right? Just look at this curve. When you have more substrate, the reaction really starts proceeding faster, right. So, what happens is one substrate typically binds to the enzyme, makes it nicer for the enzyme to bind more substrate, and when a second substrate binds, then it makes it even better for the enzyme to bind a substrate, even faster reaction, and so on, right. So, the presence of additional substrates on the enzyme enables it to bind more substrate till a point. So, n equals 4 is commonly observed. This is very classic example. So, it considers only initial rate. If you were to look at the limitations of Michaelis Menten or applying mass action, it considers only initial rate, and there is a Michaelis Menten assumption, right. So, remember that the initial rate is not the same as assuming k minus 2 is 0. K minus 2 is 0 would be an irreversible ES to E plus P reaction, that is not mandatory. Or you have the Briggs-Haldane quasi steady state assumption dEs by dt is 0, and the law of mass action itself relies on free diffusion, whereas conditions inside the cell is actually a colloid, it is not just water right, it feels more like a colloid, and then spatial homogeneity is assumed, high numbers of all molecules so that you do not have to worry about stochasticity, and remember that rate laws are approximate, they are models finally, right. So, know your assumptions, it is very important to not just start you may start the modeling exercise with some assumptions, but they will fall by the wayside once you are a few months into your modeling project, and then you may end up with very weird scenarios. Let us consider another classic example. How do we model this system? We have a system that contains 3 proteins A, B and C, where A catalyzes the dephosphorylation of B, B catalyzes the dephosphorylation of C, and C catalyzes the phosphorylation of A.
Repressilator.
So, it is not a repressilator as such. So, there is no, so all these are actually catalysis reactions, right. So, it does not inhibit. So, repressilator involves A inhibiting B, B inhibiting C, C inhibiting A. We will study that when we look at Boolean models, but here basically you have 3 phosphatase kind of system, phosphorylase system. So, A catalyzes dephosphorylation of B, B catalyzes dephosphorylation of C, and C catalyzes phosphorylation of A. So, let us try to write out these equations. So, this is C, this is A, is that right, or A and B catalyze dephosphorylation reactions. How do you model this system? What do we know? We know that dP by dt is KES roughly of this form, is that right? This is what we know. So, if you were to assume Michaelis Menten here, how would you write dAp by dt? Is that right? Well, not necessarily. This is not the back, K2 is not the backward reaction, this is actually the Km. So, let us not even write it as K2, for simplicity I wrote it as K2. So, let us call it Km1. dB by dt, remember we have conservation equations. What are your conservation equations?
A plus AP initial. Cannot be not C.
Right, which means dA by dt is minus dAp by dt, as simple as that. Okay, so, what is dB by dt here? It is going to be K2 A BP by Km2 plus BP. Then dC by dt will be K3 BCP by Km3 plus Cp. This is what we think they are. Let us look at what the paper suggested as the equations. So, we only consider the forward reaction, there will also be a reverse reaction, which is you can just think of it as a simple autocatalytic reaction, minus K dash AP
by some k dash plus a, right, k1 AC by km plus a. Right. So, dAp by dt will just be the inverse of this, right, because a plus ap is a total. So, you can eliminate one equation and basically get dA by dt is something of this sort. You do not have to use the term ap any longer, you can just call it at minus a total minus a naught minus a. So, does this sound reasonable? Does this sound similar to what we had written? Yeah, we are missing the other equation of course in both cases. See, essentially what I want to emphasize is that these equations may look complex, but if you just stare at them, they are nothing but your basic Michaelis Menten equations that we were able to write just by applying first principles, no big deal. But when you start writing this for a large system of equations, it becomes incrementally complex and can get quite challenging. So, the next step is you have now written down these equations, how do you solve them? Because what is it that we want in the end? You do not really want dA by dt or dB by dt, but you want A of t. So, how do you solve these ODEs? You now have written out the ODEs, you finally need to solve these ODEs to find out what is A of t, B of t, C of t. You have to solve them numerically, right. It is very difficult to get analytical solution to any of these ODEs. So, you will have to end up solving them numerically, and you can basically set these up as initial value problems and solve it.
In this video, we saw the classic example of Michaelis Menten kinetics, and all the modifications that can happen to Michaelis Menten kinetics when there is an inhibitor and so on. And we also looked at another example system wherein we had three proteins that or three enzymes that were being phosphorylated and so on, and what are the potential kinetic equations for those kind of interactions and so on. But the most important part is once you have these differential equations, you need to know how to solve them, as we just were speaking about, to get from dA by dt to A of t. So, that is what we will see in the next video.